Well, thank you for coming back so promptly, everybody. Dr. Jean Shanks was for many years the director of one of the UK's most successful independent pathology companies. And the Jean Shanks Lecture was the first named lecture of the Academy established by the Jean Shanks Foundation in memory of her death in 2000. And today is the 14th time that this lecture will have been delivered. Awardees are chosen on the basis of their outstanding contribution to medical science. Our awardee today is Professor Jeffery Errington FRS FMedSci, who spent much of his research career studying fundamental questions about the structure and function of bacterial cells. And he's currently the director of the Centre for Bacterial Cell Biology at Newcastle University. His work is revealing new targets for novel antibiotics, the importance of which I don't need to stress. The title for his 2015 Jean Shanks Lecture is The Antibiotic Discovery Void, Past Mistakes and Future Opportunities. Jeff, the floor is yours.
Thank you very much, John, for that flattering introduction, and thanks to the Academy as well for doing me the honour of asking me to do this lecture this year. So the first slide shows you the building that I'm very proud of in Newcastle, which is our bacterial cell biology building. And it's a very special place because it's one of the few places, if any, in the world where we've brought together a critical mass of researchers whose interest is actually in understanding how bacterial cells work. So the people who are going to do the work I'm going to tell you about tonight are shown here, and I'll try and remember to acknowledge them as I go through the lecture. So, as I say, this is called the Centre for Bacterial Cell Biology, and it reflects my research interests over the last nearly 30 years. So, what is bacterial cell biology? Well, it goes back, the discipline, if you like, goes back about 20 years now, and it was founded really by two technological advances. One which has been particularly important over the last 20 years, was a revolution in digital fluorescence imaging. So about 20 years ago, it became possible to actually look at the substructure of bacterial cells in unprecedented detail using fluorescence imaging. And just to give you, there are many thousands of these images, but just to give you a couple of examples here, here's a bacterial cell. You can see how small they are. This is 1 micron. So these are actually very close to the resolving limits of the light microscope. And then the red here illustrates the membrane of the cell, or pair of cells, and the green blobs here represent the chromosome stained with a different color. And so you can get these sorts of images started to give us indications that bacterial cells were much more structured than had hitherto been realized. But in fact, if you look at the image on the left, when we started to be able to label individual bacterial proteins, we found that the localized structure of these cells was actually remarkably sophisticated. And so here's one example of a protein that we've looked at probably about 10 years ago now. It's actually homologous to actin, which is a major cytoskeletal protein of our cells, eukaryotic cells. But in bacteria, it also has a function in cell shape. And not only that, it polymerizes to form filaments, which are very similar to actin filaments. And these filaments wrap themselves around the periphery of the cell, and they're actually involved in determining the shape of the cell. So these cells are rod-shaped because they have this MREB protein actually directing the shape. And many hundreds of proteins that have been localized in bacterial cells have turned out to illustrate amazing substructural features in these cells. So it's really completely transformed our view of bacteria, which had previously, I think, been viewed as bags of enzymes, into things which are incredibly structured. The second thing that happened is that whole genome sequencing and next generation sequencing came along, and this has absolutely transformed bacterial molecular biology again, because bacteria have very small information-rich genomes, and so the DNA sequencing has really been very influential moving the field forward. And if you ask what this research is for and why I'm actually a member of the Academy of Medical Sciences, well, there are, I think, two important reasons. One is that the research underpins antibiotic discovery. So you need to understand the structure and function of bacterial cells if we're going to be able to develop new classes of antibiotics in the future. And secondly, the basic understanding that we get is perfectly relevant to understanding of disease organisms, organisms that cause disease. So I probably don't need to introduce this audience to antibiotics, but I'm very fond of some of these images because I worked in the Doesnt School of Pathology for about 13 years in Oxford. And of course, everyone knows that Fleming discovered penicillin through this serendipitous observation back in the 1930s, and then Flory and Chain and their team succeeded in purifying this molecule penicillin. And the advent of antibiotics arguably is the single most important advance in medical science, period. It transformed our ability to treat infectious diseases virtually overnight. And I've just got one slightly amusing image here to show you what happened in the 1940s. People suffering with these infections, which had been previously virtually untreatable, could now be cured in 4 hours by the administration of one shot of penicillin. And the same applied to people suffering from pneumonia, which had a very high mortality rate beforehand. So antibiotics have really transformed medical science. But those with good eyesight will see that I've put a question mark against the antibiotic era up here, and that's because, as you all know by now, there's a serious threat from the emergence of antibiotic resistance. And if you look at you only need to go to the internet or watch the news to see images like this. This is taken from the BBC website a few days ago. Some bacteria are becoming resistant to our best drugs. And it's not just the BBC. There are very important and influential people who are making statements about this. If we fail to act, we're looking at an almost unthinkable scenario where antibiotics no longer work. We go back to the dark ages where treatable infections and injuries will kill. This is the Prime Minister last year. And of course, our own Dame Sally Davies. I'm not sure if Sally is here at the moment. I think she's coming later on. But Sally has really pushed the vote out, if you like, to try to get antibiotic resistance back onto the map in her reign as the government's chief medical officer. And so she compares in this quote the threat arguably as important as climate change. And I think in another quotation, she's compared it also to the global threat of terrorism. So this is a really, really serious problem for us. You could call it a catastrophe. So what's happening? Well, resistance is increasing to virtually all known antibiotic drugs. The resistance to last resort antibiotics is emerging. And just 2 weeks ago, I think there were reports from China of resistance to colistin emerging, and it's on a mobile genetic element, so that can spread to other bacteria. This is not a terrific antibiotic. It's actually quite toxic, and it's used as an agent of last resort for people that have, for example, urinary tract infections in hospitals. And so if we lose colistin, we really are in very serious trouble. There have been no new classes of antibiotic drug discovered and marketed since the 1980s. And if we start today and we look for a new target, typically it takes between 10 and 15 years to get the molecule onto the market. So there's a long lag time before we can get new antibiotics out there. So what happens if we go back to the pre-antibiotic era? Well, most of us in the audience are probably too young to remember this, but people would pick up what we would now call common infections from gardening or strep throats, and they would die from these infections. There was no treatment for that. And you can see that back in the USA, pneumonia was ranked third as a killer in the pre-war era. Elective surgery, hip replacements or knee replacements, and a number of us probably starting to think about this kind of elective surgery at this sort of age, becomes impossible if we don't have good means of treating infection. And any diseases which involve immunosuppression, so HIV obviously, but also people who are undergoing transplantation or many cancer treatments have become immunosuppressed. And the only way we can keep those people alive right now is by using antibiotics to prevent them from going down with serious infections. Tuberculosis, pneumonia, sexually transmitted infections, whooping cough, all sorts of diseases would kill people in large numbers before the antibiotic era. So what are we going to do about this? So I thought I'd give you one slide just to illustrate the process that we use, or pharmaceutical companies have used in the past, to try to find new antibiotics. And so this is a simplistic primer, if you like, for antibiotic discovery. And so I see, from my perspective on this, three steps. These two are very early steps, but I think this is where the real problem actually lies. You need to start with compounds, chemical compounds, usually large collections of compounds, And then you have some kind of screening assay, which means that you look through the compound collection to find molecules that have the activity that you are interested in. And then you take these molecules and you put them into this hugely expensive and time-consuming process called drug discovery and development, many hundreds of millions of dollars, 10, 12 years, to take those molecules and get them actually onto the market. But I believe that this is really where the problems are at the moment with antibiotic discovery. So let's take a closer look at this, about how this is done in the antibiotic arena. So you start with either small drug-like molecules that have been made by synthetic chemists, or natural product molecules that are made by organisms. And typically, in the past, pharmaceutical companies would use what we call a kill assay. So it's a very simple assay. You take something like Staphylococcus aureus, and you look for chemicals that will actually kill the bug. So you look for things that are potentially antibiotics. You get many hits, so you find many molecules that will successfully kill Staphylococcus aureus. The problem is the vast majority of these molecules are actually also toxic. They kill human cells just as well, if not better, than they kill the Staphylococcus aureus. And secondly, you keep on rediscovering, among the things that are selective, the same old molecules, tetracyclines, beta-lactams, vancomycins, and antibiotics that had already been found. So about 20 years ago, the pharmaceutical companies, en masse, really switched the way they did antibiotic discovery. And this actually aligned with the way they did discovery for other kinds of therapeutic molecules, to a thing called a targeted approach. And so for an antibiotic, the secret to targeting is that you choose a target in the bacterium, which is essential for the bacterium. So if you inhibit it, the bacteria will die. It should be different from the previous antibiotic targets, so you find things that are, so you get around this rediscovery problem and find novel molecules. It has to be absent from humans. So it's a target that's a special target that's found in bacteria but not in humans. And the idea of this is that you get selective toxicity. You kill the bacterium, but you don't kill the patient in doing so. And then finally, it should be druggable. The molecules should have some of the properties that would make it look attractive from a drug development point of view. So this is an interesting approach. In principle, it works very well. You should find novel compounds because they should be acting on different targets. Toxicity should be low because it's a specific target to bacteria, not non-humans. But there are two big problems. The first is it's very difficult to find these molecules. So the frequency at which you get something which hits a novel target, which is, well, in principle, and actually which works on bacteria, is pretty low to vanishingly small, depending on the target. And secondly, as I'll show you in a minute, there's an intrinsic problem with this approach, which lies in the evolution of resistance. So I've been interested in trying to develop antibiotics for probably about 20 years now. And again, back when I was in Oxford, we got involved in antibiotic discovery. We set up a company called Prolysis, which set about trying to find molecules using this targeted approach and looking at some of the functions that we had a real academic interest in. So in this example here, we were successful actually in developing compounds that specifically inhibit cell division in bacteria. So this is what Bacillus subtilis, my favorite organism, looks like when it normally grows. So the cells elongate, then they divide in the middle, and they do this with incredibly high fidelity. So starting with a single cell, you can get to many thousands of cells within a few generations. However, if you treat these cells with our cell division inhibitor that Prolysis developed, then what you see is that the cells can continue to grow for a while, but they don't divide. And then they become unstable. And if you watch just the end of the cell here, you'll see that the cell actually explodes at the cell tip, which is probably the site where the last cell division occurred after the cells had been treated with a compound. So they become unstable and they lyse. So these are very effective molecules at killing B. subtilis. They also kill pathogens. So in a paper which we published in 2008, we were able to show that this molecule, one of these molecules, a benzamide derivative, succeeded in, would also inhibit cell division in Staphylococcus aureus. And not only that, we can actually show that the administration of this molecule to mice that had been challenged with a lethal dose of Staphylococcus aureus could be rescued by administering this molecule. So we could completely rescue the mice by treating them with the benzamide. So what's the downside of this? Well, unfortunately, it turns out that this benzamide, like a lot of the molecules that industry were trying to develop, binds into a very specific pocket in its target molecule, which is a protein called FTSZ, a bacterial cell division, critical bacterial cell division protein. And what happens is, as soon as you start to use a lot of the compound, you start to select for resistance mutations. And what happens is these mutations arise by changing these amino acid side chains in the binding site for the benzamide, such that they block or occlude the insertion of the molecule. And that prevents the molecule from working. And some of these mutations, although some of those mutations are actually deleterious, they affect the function of the protein, others leave the protein completely unaffected. And so because of this resistance problem, which is a very common problem for compounds that target single lethal enzymatic targets like this, pharmaceutical companies were very reluctant to actually take on the risk of developing these compounds and spending hundreds of millions of dollars if it was possible that as soon as they came onto the market, resistance would emerge. So targeted small molecule approaches have largely failed, and it's been a very expensive learning process, particularly for Big Pharma. ProLysis spent quite a lot of money on this, but Big Pharma spent hundreds of millions of dollars in this. So here's a review from GlaxoSmithKline in 2007 describing their experiences with this approach. And here's a couple of sentences from near the end of the review. They looked at 300 genes. They ran 70 high-throughput screening campaigns over 7 years, hundreds of millions of dollars. And at the end of it, the pipeline of novel mechanism antibacterials is still empty. These guys are, well, some of these guys still have their job. I find it quite surprising. Maybe I should have said that in public. So AstraZeneca have a similar experience. So here's a much more recent review. Again, the bottom line of this approach, of this review, is that despite huge amounts of effort over many years, the researchers have struggled to identify new small molecules with meaningful cellular activity, particularly for gram-negative bacteria, gram-negative pathogens. So this has been a really very disappointing experience for many people. There is one kind of light on the horizon. And so here, it turned out that Merck were also interested in our benzamide antibiotics, and you can see that they put a lot of people onto this problem. And what they were able to show was that there was a possibility of using benzamide with another antibiotic, an imipenem, a beta-lactam antibiotic, because these antibiotics actually turn out to work synergistically. So if you administer the two compounds, you get a better effect than if you use either of the compounds singly. And so that's one way of avoiding resistance, because even if resistance occurs to the benzamide, the cells will still be killed by the imipenem. And this kind of approach has been used quite a lot in antiviral therapies, where it's quite common to use combinations of drugs. So maybe this is a way to overcome resistance problems, but surprisingly, Big Pharma seem reluctant to use this approach in the antibacterial arena, even though there is one very good example of this in Augmentin, which has been a very successful drug. But it seems that pharma are really quite reluctant to use this kind of approach, at least at the moment. Maybe in years to come, we'll be forced to actually start to think about this kind of approach. So what are we going to do about this? Well, in the last few years, we've become very interested in returning to a wonderful source of therapeutics. So natural product drugs from bacteria. So historically, these were a major source of antibiotic leads. Not just antibiotics, in fact, but many other kinds of drugs. I mean, surprisingly, these bacteria, like this beautiful actinomycete, which I show over here, also make things that are really quite surprising. They make statins, immunosuppressants, anti-cancer drugs, and antifungals. Probably the reason they make these molecules is that they live in the soil with huge numbers of competitors, both bacteria and fungi, and even worms and small animals. And probably they make these molecules to actually counter the threats from the things that they're competing with in the soil. So they've been a fantastic source of drugs in the past. They have fantastic structural diversity, much more structural diversity than the medicinal chemists will ever achieve. And so here are some examples again. An anti-cancer agent, an immunosuppressant, rapamycin, an antifungal, and vancomycin, one of the last resort antibiotics that we have. You see the really complicated structures of these molecules. So there are two reasons why these molecules are particularly attractive as sources of antibiotics in my view. And they both stem from the fact that they have a very sophisticated mode of action which has been selected over evolutionary time. So these molecules wouldn't have survived in nature if they weren't useful to bacteria living in soil and among these the myriad of competitors. So the first thing that they do, which is different from the approach that I've described, is that they have multiple lethal targets. So they hit more than one protein that's essential for the cells. So the beta-lactams, the penicillins and cephalosporins, there are multiple penicillin-binding proteins in bacterial cell wall machineries that are all essential for the viability of the bacterium. So you can't get a single point mutation generating resistance to a beta-lactam. Secondly, things like vancomycin do something really clever, and that is they bind to a non-protein target. In this case, they bind to the cell wall precursor molecule. So it's a small disaccharide molecule. And it's very difficult for cells to escape from that because they need to change the structure of the small molecule that's used as a substrate for cell wall synthesis. And then they'd have to change all of the enzymes in the cell that normally interact with that substrate to either make it or to assemble it into the cell wall. So again, you can't get simple point mutations generating vancomycin resistance. You need an array of something like 5 or 6 genes to be acquired from somewhere else to become resistant to vancomycin. So it's a very successful drug from that perspective. Unfortunately, pharma companies largely dropped their natural product discovery in the 1990s, and there were really 3 reasons for this. First of all, is that rediscovery problem that I told you. You keep on finding oxytetracycline and streptozotocin and vancomycin and those molecules. Secondly, the medicinal chemists hate these molecules because they're structurally very complicated. It's very difficult to make analogs of those molecules that have many chiral centers. And thirdly, there was an alternative approach called combinatorial chemistry, which in the late 90s became a preferred mechanism for generating chemical diversity at around about that time. And so chemists would make libraries of closely related compounds using methods that enabled them to make many hundreds or thousands of related compounds in a short space of time. Unfortunately, the amount of chemical structural diversity you can get with combinatorial chemistry is nowhere near what you can achieve by looking for natural products. Okay. So I believe, and I think a lot of people are beginning to agree with this, that there should be or is already a renaissance in looking at natural product molecules. And there are 3 big reasons for thinking this is a good idea. First of all, there's a big change in our understanding of the diversity of the organisms that make these molecules. So many new sources of natural products have recently been discovered through what used to be called non-culturable organisms, so organisms that are very slow growing or very difficult to grow. We now understand how to isolate those and to grow them. And also now it wasn't realized until relatively recently that actinomycetes aren't just found in soil, they're also present in huge numbers in marine sources. And so there are lots of diversity coming from there. Secondly, there have been big advances in chemistry since the late 90s. Purification technologies have improved, so it's much easier to purify compounds. And the analytical methods have advanced as well. So rapid and sensitive mass spectrometry and NMR methods have made it much easier and quicker to identify and determine the structures of complicated natural products. And finally, genomics has transformed our understanding, as I'll show you a little bit later. We now know that there's huge untapped genetic potential in the organisms like this that make our natural product molecules. Probably we've only been looking at the tip of the iceberg of the capability of these organisms. So for every antibiotic that we know that these guys make, there are probably, from the genomics, predicted to be 19 or 20 other molecules that they can make. Because these are biological organisms, if you like. They've been selected by evolution. We'd imagine that all of this coding capacity is for making molecules that are biologically active, complicated molecules that are biologically active. So it's just a question of figuring out what those biological activities are and trying to harness them. And also, we can use these genetic methods to make analogs of complicated natural product molecules as well by playing around with the genes that actually are responsible for making the molecules. So Another reason why I was influenced in this was because a guy, Mike Goodfellow, who had taught me microbiology as an undergraduate at Newcastle 30-odd years ago, retired very recently, but I hadn't realized at the time, but he's the world-leading expert on the taxonomy of the actinomycete bacteria, the things that make most of these natural product molecules. So he spent his whole career, 40 years or so, collecting these organisms. Look, he's been all over the world to deserts and mountains and marine trenches to look for these organisms. And of course, he's driven by identifying novel organisms that no one's found before, finding new species. And so he has a beautiful collection of about 10,000, which is a relatively small collection, but it's very well dereplicated. So there are very few replicates of the same organism in this collection. So this makes for very efficient screening from a drug discovery development. And our prediction, and I think a lot of people would agree with this, is that it's likely that if you look at novel organisms, you're going to find novel chemistry. And I think this is starting to be borne out now. So what we've been doing, just very briefly, or Demiris has been doing, is to take this Mike's collection, they've been curating it and screening it very simply using the traditional kill assay for things that would kill a gram-positive bacterium or a gram-negative bacterium or yeast. You can see that there's huge antibiotic coding capability in these organisms. So 28% of them make a molecule that will kill gram-positive bacterium. This is a huge problem because it's very difficult to know what to do with the 2,000 hits and where to, how to get around this dereplication problem. With gram-negative bacteria, however, it's a manageable problem, but you can see that these things are much more resistant. And this is because gram-negative bacteria have an extra outer layer that protects them from the action of antibiotics. Nevertheless, there are a little over 150 organisms that are making things that will kill E. coli, and the company is gradually working through those. And the results are still very preliminary, but there are exciting indications that there are going to be a number of new different molecules that are capable of killing gram-negatives. And the future will tell whether they turn out to be useful as drugs. There are also nearly 1,000 potential antifungals here, which they're working on. So these, of course, are the highest priority. But since there wasn't a great deal I could tell you about that screening approach, what I thought I would do in this talk, and this latter part now is, this is really unpublished data that I'm going to tell you about, is a sort of new old approach to trying to find novel antibiotics. And so we've been interested for a number of years now in using a process called chemical genetics to screen for interesting molecules. And so my background originally is a geneticist. And so for much of my career, I've been trying to use genetics to dissect out understanding of the function of bacterial cells. And so the way this works, for those of you who don't think genetically very much, is that you start by taking cells and you randomly mutate them. So you introduce mutations randomly into the chromosomes of the cells. And then the big trick and the quality by which geneticists in general are judged is by how clever the genetic screens are that you use to find mutations that affect a particular function. And so you screen for mutants affected in a specific function. If you find them, then you know that the mutations lie in genes that are involved in that function. That's basically how genetics works. But about 20 years ago, we realized that you could use a very similar approach. This is actually before the term chemical genetics had been invented. You could take a really good genetic screen and actually use it to look for chemical compounds that would elicit a similar kind of effect. So you can use a genetic screen to look through collections of chemical compounds for things that have a particular effect on cells. And by doing this, you hope to be able to find compounds that have a very specific mode of action, hopefully on interesting novel targets, and actually to do this in vivo, actually on cells. So it's ideal for natural product screening, particularly for antibiotics, and the products of this could have both academic and commercial use, as I'll show you. So most of our effort has been targeted at the bacterial cell wall, because this is really the ideal target for antibiotics. We really don't yet have a better target for them. The wall is essential for growth and viability, and the reason why it's particularly useful is that bacterial cells have a very high internal osmotic pressure, which pushes out on the cell wall. And if cell wall integrity is breached, then the cells explode. And this is why antibiotics that act on cell walls are particularly effective. There's no equivalent of the wall in human cells, so you get very good selective toxicity. And the best of our antibiotics, particularly beta-lactams, penicillins, and cephalosporins, also vancomycin, mentioned, these are still our best antibiotics. But there is potential for new classes of antibiotics. There are a number of enzyme activities associated with cell wall synthesis that have never been exploited so far as antibiotic targets. So just to show you what these beta-lactams like penicillin do, this is a movie made by my friend Ken Gerdes before he left us at Newcastle, showing you E. coli cells treated either with penicillin or the controls. What you see is these cells basically, these cells, the control cells, of course, grow and divide quite nicely. The cells on the left, you can see they undergo this very spectacular cell lysis. So they basically pop. And so these are exploding because of this high internal osmotic pressure and because they have lesions in their cell wall. So this is one of the reasons why these antibiotics are really, really very, very effective. So what can we do to take advantage of this? So we set up, and this is a really, really great collaboration between my academic lab and the guys in Demuris. We set up to do chemical genetic screens for inhibitors of 3 different cell wall targets. And I'm just going to tell you today about one of them that we call the missing glycosyltransferase. So the glycosyltransferase is an enzyme that's very closely related to the target of the beta-lactam antibiotics. So the beta-lactams interfere with an enzyme called the transpeptidase, which is one of the enzymes that makes the key bonds that make the cell wall peptidoglycan molecule. But most of the enzymes that have this transpeptidase activity have a second enzyme activity called glycosyltransferase, which is involved in stringing together the glycan strands that are then cross-linked by the transpeptidases. So these enzymes, in principle, are equally good as targets for antibiotics. But about 10 years ago, we were astonished to see this report from David Popman's lab about Bacillus subtilis, our favorite organism again. We knew that this organism had 4 glycosyltransferases. We assumed that these would be essential for viability, but David showed that he could knock out all 4 of those genes, and it made no difference whatsoever to the viability of the cells. They still seem to make cell wall perfectly normally. And in fact, 10 years of effort in my lab and a number of other labs around the world have failed to identify this missing glycosyltransferase. It must be there because these bonds are being made to generate the peptidoglycan, but the cells don't have the conventional glycosyltransferase. So it's a very interesting potential drug target. And so we wondered whether we could maybe find it by chemical genetics. And so our prediction was that if we took this mutant with the 4 genes knocked out, should be hypersensitive to inhibitors of the missing enzyme. Okay? So the first thing we did was we confirmed David's observation that if we make this quadruple knockout, that actually, indeed, those cells are viable. And then what we did was we screened just over 2,000 actinomycete extracts for differential activity on this mutant compared to wild-type strains. And we found initially about 42 candidates that looked as if they were differentially active in killing the mutant compared to the wild-type. We retested those in detail. So there's a typical kind of drug screen. 3 of them turned out to be readily confirmed. So we repeated the growth in the extract preparation. We got the same result again. And so far we've prioritized this actinomycete strain called 20654, which makes these beautiful colonies for further work. So this gives me an opportunity to tell you about the genomics of these actinomycete organisms, because they're really amazing bugs in a number of ways. So we've got the genome sequence now for this favorite strain, 654, and it has a genome of just over 8 million base pairs. So this is about twice as big as E. coli or Bacillus subtilis. And the sequence is good. These organisms are amazing in that they have very high G plus C content. 72% of their DNA is Gs and Cs, rather than As and Ts. So it's very unusual. We don't understand why these organisms have this high G plus C. But there are then programs like this one, Anti-Smash, which was developed by Enrico Toccano now at Manchester, that can analyze the sequence to find the gene clusters that are responsible for making these natural product molecules. And so when you run this Anti-Smash program, it tells you that this organism has got 49 recognizable natural product gene clusters. And the program will tell you about these. There are 8 different polyketide molecules predicted. There are 5 non-ribosomal peptides, 4 terpenes, 7 mixed polyketide terpenes, etc., etc. And these modules, if you like, are scattered around the chromosome of the actinomycete, and they comprise something like a quarter of the genome. So a quarter of this bug's genome is devoted to making natural product molecules. And we know that from our work that it makes at least 3 antimicrobial molecules, so the one we're interested in, and also an antifungal, and also a non-specific antibiotic. But we have no idea what the other 46 natural product gene clusters are in this organism. And this is the same for all of the many other tens, or hundreds of thousands, or millions of actinomycetes that are out there in the soil and the sea. Okay, so one of the things we'll try to do in the future is to identify which of these clusters is responsible for our molecule, and then we can start to use genetics to dissect how that molecule works. The really difficult part of this whole process is the purification. And so this just gives you a kind of overview of how the process works. You start off with a big culture of the actinomycete. You can see it has a beautiful red color. You use these beads to absorb the organic molecules. Then you can use things like TLC, very old-fashioned methods to explore which solvents to use, do solvent extraction, and then a series of chromatographic methods that you can use to separate the different molecules that are in the extract, because there are many, many different molecules in these extracts. Eventually, you get to HPLC trace with peaks, and you can test those peaks individually to see whether they contain the molecule that you're interested in. Eventually, you can demonstrate the differential activity and use mass spectrometry on the purified molecule to get a very accurate mass for that molecule, from which you can figure out whether this is probably a new molecule or whether it's something that's already been discovered and published in the past. And so we have, in this particular case, we know the accurate mass of this molecule. It looks as if it's different from things that have been described previously. It's about the same size as penicillin, so it's in the right small molecule range for potential development as a drug in the future. Does it work? So here's the proof in a sense. Here is the pure compound now from that process that I've shown you. If you compare the wild-type B subtilis and the mutant B subtilis, you can see that the compound is much more active on the mutant strain than it is on the wild-type strain. And here's a control compound just showing that this is not just a sort of an intrinsic difference between the mutant and the wild-type. This is really a differential activity on this particular mutant compared to the wild-type. So we're very excited about this molecule, but what does it do to cells? Well, here's an experiment. So these are still preliminary experiments at this stage, but beginning to become very exciting, because what you see here is if you take the wild-type cells, and then we take the 4X mutant with these genes knocked out, in the absence of compound, they look fairly similar. This has it has a slight morphological difference in the wild-type, but nothing significant. But in the presence of compound, you can see that this strain is undergoing dramatic degree of lysis. So you get these roundish, phase pale cells, which are basically lysed cells. So this compound seems to be doing what we would have predicted it should do if it's inhibiting the missing glycosyltransferase that we've been hoping to find for about the last 10 years. And also, potentially, we have a compound here which in principle, could go into discovery and development as a potential treatment for certain kinds of infections. Obviously, we're a long way from that so far. Finally, I thought I should show you a picture of Carvey, because he's an incredibly hardworking and diligent postdoc. He hates this picture, but he refused to give me another one, so I've showed it to you anyway. But he's looking a little bit somber here, and he's somber because this is another compound, a different strain, one of the other candidates that we had. And what you can see is that, well, first of all, I should say that this one is even more difficult to purify. The strain grows incredibly slowly. The molecule is difficult to purify, but we're excited about this one because it really looks very specific for the 4X mutant rather than the wild type. It doesn't seem to have any effect at all on the wild type, whereas it does kill the 4X mutant. So this is in progress. So I could tell you stories, similar stories for the two other enzymes that we've been looking at, and we're really getting very excited now that this kind of approach looks as if it's actually starting to work. So we have in the case of this lipoteichoic acid synthase, we have active compounds that are partially purified. We know the structure of one of them. It looks like a substrate. It could be a substrate analog for this enzyme, and it's active on Staphylococcus aureus at least, given the quantities we've managed to purify so far. And we have another story for the wall teichoic acid peptidoglycan ligase, so another late step in wall synthesis. And we've got several extracts that clearly differentially affect the cells that have got mutations in some of these genes. So I'm not sure how I'm doing for time right now, but I've come to, I think, my summary slide. So I think there are a couple of messages from this that I'd like you to take home tonight. The first is that, of course, as you all know, there is an impending catastrophe in terms of antibiotic resistance. This is going to be a global problem. It needs to be something that governments all over the world actually collaborate on. Even if we start now with projects like the one that I've described, it will be a decade or more before those molecules get to the clinic. So we need to start doing things right now. We need to improve funding situations for discovery and development. Combi-chem approaches, the targeted approaches, have really failed to deliver in the last 10 or 15 years. I think the natural products are one area that we should revisit. There are good reasons for actually deciding to do this in terms of improved chemistry, microbiology, and genetics, genomics. I like the idea of using these chemical genetics screens, although the real problems that I haven't talked about today are these gram-negative pathogens. All of the screens that I talked about there that have worked for us so far are actually to do with gram-positive bacteria. We really need to make dramatic advances and find molecules that are active on gram-negative pathogens, because that's really going to be the biggest problem in the next few years. But potential new antibiotics are emerging, so maybe there is some light on the horizon if we continue to pull together and work on this. So that's, at that point, I should stop and thank the people who really did the work. None of this work was done with my hands. I'm indebted to a fantastic team of people. You see, there are some sunny days in Newcastle in the northeast of England, so this is my academic lab. Sat outside our Centre for Bacterial Cell Biology. I'm grateful to the ERC and the Wellcome Trust for funding that work. These are the guys from Demuris. Some of that funding came from Innovate UK. And I'd like to stop there and thank you all for your attention, and again for this wonderful award.
Jeffrey, thank you for a wonderfully clear lecture, and I think also for giving us hope. So Jeffrey's kindly agreed to take questions, so please, there are roving microphones, I hope. Please take a question there.
So I was just curious, did your forward genetics screen identify your missing glycosyltransferase, and if so, is your new compound actually targeting it?
Yeah, it's a little bit too early to tell at this stage. So it's really all of the data that we've got I've actually shared with you. At the moment, we don't have enough compound to do the sorts of experiments that you're alluding to, because we can do the microbiological experiments, killing cells and looking at the morphology, with rather tiny amounts of compound. But once you start to need to do biochemical assays binding the compound to proteins, for example, we need to make a lot more of the molecule. So where we are now is scaling up the process so that we can actually get our hands on milligram quantities of the compound.
There's a question, gentleman here.
Yeah, so in your chemical genetic screens, that's would be equivalent to getting synthetic lethals. So in that case, you're aiming towards combination therapy to have these to be effective in use. Right. And in which case, then you have to really convince both the industry and in clinical practice to be thinking about. So, in terms of that approach, it just seems that it seems a harder route to go down.
I think I haven't had time to tell you a lot about some of the other screens. So, for example, some of the other screens that we have aren't based on that kind of synthetic lethal. The second thing is that Bacillus may not be the ideal organism for doing these kinds of tests because it's also a soil organism. So it has, for example, 4 of these glycosyltransferases. It has 3 of the MREB proteins that I showed before. It has 3 or 4 lipoteichoic acid synthases. Probably it uses these because it's subjected to attack by other actinomycetes in the soil. So our impression is that it's still possible that with pathogenic organisms, which often sort of trim down their genomes, that some of these molecules may actually work directly on those organisms without the need to have a combination therapy. So again, when we get a bit more of this molecule, we'll start to do some detailed microbiology on those organisms, on the compounds, and it may well be that we'll be able to find organisms that are susceptible because they rely on that other glycosyltransferase for viability. So it's really an illustration of the sorts of approaches that we're using. But now, of course, we can get at that glycosyltransferase because we can look for resistant mutants, for example, to try to find the gene. Hopefully we can finally get this gene after 10 years.
I've got perhaps more in the nature of a comment than a question. I mean, the problems of antibiotic resistance, I remember reading papers by Demeritz going back to 1948 and the obvious answer of using combinations, and then that was killed by the fact you could get multi-resistant plasmids.
Sure.
So one question is, do you think that it's going to be possible with some of these newer approaches you're using, which are really very elegant, that one will get the same problem? My comment was that in a strange way, where bacterial genetics led the field in so many ways, you're now going back to an approach that those of us who work in cancer have thought was what we should be doing anyway, which is to study in more detail the genetics, genomics, call it what you will, of the organism, to find novel targets that in other ways than the ones you mentioned to start with, which I think is an interesting issue, and also the power of the using evolution, which gives you a much stronger result than you can ever get in the laboratory.
Sure. I think I agree. I think the resistance issue, it's always going to be a problem. I don't think there will ever be an antibiotic that we will not
Do you
get
think they'll
resistance
accumulate on plasmids the way that the conventional antibiotic resistance does?
I think they will. And, I had a slide. I wasn't sure how much time I would have. I had a slide of kind of recommendations, and one of the things that I feel really strongly about is, we worry about prescribing antibiotics in the UK, over prescribing, but actually the real problem is in underdeveloped parts of the world where they throw huge quantities of antibiotics into animal feed or even into the sea, where they have prawn or shrimp farms. They'll dump huge quantities of ciprofloxacin, for example, into the sea water to improve the yield. So that's, I think, where the major source of resistance is going to be. And you see colistin, nobody ever thought that there would be resistance to colistin, but now it's starting to emerge in China, and it seems it's because they're using huge amounts of this stuff in animal feed.
Okay. There's 2 questions at the front here. Take these 2. George?
Hi. We keep being told that Big Pharma doesn't want to develop new antibiotics because there's no market for them. And, the reasons for that are quite familiar. What does that mean? Does that mean the state or public bodies or charities have to pay for the whole drug development of any new compounds?
I think there are a lot of solutions to this problem. Again, it's a shame Dame Sally isn't here because she'd be well placed to comment on that. And I think there are various things that we can do. One, the US government has actually done quite a lot to try to convince pharma companies to come back into the market, by, I think there are at least 4 different acts that have been put through that actually, address some of those issues. But I also think, it's going to have to be some kind of public-private partnership in the long run because, pharma companies, because it's so difficult to develop antibiotics and it's so expensive, it's going to require a different funding model, I think, in the future.
Patrick, would you enlighten us? I know you've thought a great deal about this.
Thank you for your carry on the theme of hope you had at the end. I thought you might like an update on the people that you thought might have lost their jobs in 2007.
I should have realised there'd be somebody from GSK here. So.
David Payne, who was the first author of that paper, heads antibiotic drug discovery at GSK, and he has actually got the first of a new class of antibiotic which hits two mechanisms simultaneously in late-stage clinical development in multiple different diseases, including drug-resistant gonococcus and gram-negative organisms. In fact, it works against colistin resistance and also plague and other bioterrorist threats. So I think,
okay, I wasn't
there's a lot of effort goes into these areas, and it's perseverance in part which gets there. I do think hitting two targets is quite important. Hitting two targets at once is quite important in terms of the resistance profile.
And the financial models, Patrick?
Well, I think we've persisted with it on the grounds that this is something which is needed for society, and there will be a financial solution to this somewhere down the line. And my own view is that the last thing that society needs is a company to make a new antibiotic and then market the hell out of it. Yeah. I mean, you actually want to reserve it, and that's really where the challenge comes. And I think there are models which are being explored now which address that, which I think is a very important thing to address.
Apologies to David.
Got time for perhaps one more question or any more? Yes, please, George.
Time is of the essence, clearly, for human use. In the molecules that you're producing, do you have parallel screens on human cells, say hepatocytes or whatever, so you could see toxicity very quickly?
Sure.
I mean, there are very few drugs that have no toxicity, of course, depending on dose.
Sure.
Are you doing that as well in parallel?
It's a key early step in the process, actually demonstrating that, or at least testing for toxicity and making sure that there's a big therapeutic window between difference between the toxicity.
So would you abandon a drug that was toxic, or would you pass it on to try and do some additional chemistry to alter it?
I think it would be it would depend on the molecule, I think, and the extent to which you could differentiate between the activity on the bacterium and the activity on human cells. So, if you've chosen the target carefully, then toxicity should be less of an issue. But it's always a very key early step. And I think my experience, maybe Patrick would say the same thing, is that it's actually very difficult to engineer out the toxicity if you have. Often it's telling you that the target is intrinsically a bad one.
But as you say, that's why the bacterial cell wall is such a good target.
Yeah, absolutely.
I think we must close now, but I can't do so without thanking Jeff again for a splendid Gene Shanks lecture. Thank you so much. And in recognition of which we have the science certificate. Jeff, congratulations. Thank you very much indeed. Thank you. Good. So now that brings the session to a close. There is a drink reception for all attendees. And then if you're fortunate enough to have a blue sticker on your badge, join us for dinner. Dinner will start at 6:15. Thank you very much indeed.
I thought I should add a postscript to my lecture based on the comments made by Patrick Vallance of GlaxoSmithKline near the end of the questions. He was rightly pointing out that GSK have recently been running phase 2 clinical trials on a novel antibiotic which looks very promising. I was not aware of this program before the lecture. If this compound makes it through to the market, it'll be perhaps the most important development in the antibacterial field this century. Nevertheless, the compound that's being discussed belongs to a class of molecules that has actually been in development for nearly 20 years, and it acts on a traditional target, DNA topoisomerase. One completely new antibiotic would be a step in the right direction, but we are going to need an array of new antibiotics with new modes of action if we are to stay ahead in the battle against resistant bacteria.